Okay, for part C, we're going to continue the parasite and host conversation, looking at a couple examples of coevolution. So, not shockingly, when you have this much selection happening by both the parasites to do well in the host, and then the host to get rid of the parasites, you tend to have this coevolution. It kind of results from the arms race as well, right? So when a parasite and a host each possess specific adaptations, it suggests that the species have undergone evolution. Coevolution occurs when populations of two interacting species evolve together, each in response to selection pressure imposed by the other. Parasite attacks a host. Host figures out a way to defend themselves from that parasite. Parasite evolves a way to circumvent the host's adaptation, so on and so forth. So arms race is kind of an effective way of thinking about it. This is kind of a crazy example. This is from Myoxma, which is a virus, a lethal virus in rabbits. And, Australia has this long history of invasives and this long history of adding an invasive to kill the other invasive. And we looked at one a couple chapters ago that actually worked. It was the moth and the cactus. There's many more that don't work. And so here, I forget what the initial number was. I thought it was like in the 40s or 50s. Numbers of rabbits that were introduced to Australia quickly grew to I want to say it was hundreds of thousands, if not more. Turns out rabbits breed like rabbits. And so to deal with this massive rabbit population, they decided, hey, let's release a virus. That's a good idea. And so they released this Myoxma virus to Australia. And it was, shockingly effective. It killed, about 90% of or more, maybe it was 98% of the rabbits. But then over time, you start to get rabbits that are genetically resistant to the disease. And so then you got to rebound the population back after that. There's a local rabbit disease that's kind of scary. It's not Myoxma, though. I just looked it up. I thought it might be. I don't have rabbits, but I know some folks on the San Juans that do, and there's a lot of concern about that because they have pretty high feral rabbit populations out there. They're non-native introduced rabbits, and so there's always this concern about having a wild rabbit have a disease and pass it to a domestic rabbit. It's something that's a hemorrhaging disease. It's different. It's not caused by this virus, but it's kind of a similar pathway or similar symptoms. And so we can look at the rabbit mortality rate, 100% being 100% mortality, and then the number of epidemics experienced by the population. And so if you have a population that's experienced 7 epidemics, the mortality rate within that population is quite low. This graph starts at 20%. You should never start a graph at 20%, but this one does. So that's 25% or so of the population would die from the epidemic. In a population that's only experienced 2 epidemics, you're closer to 90%. So there's still an impact from it, but much less. This is the data from Australia. Which graph is this? B. Determined against. Okay. And this is kind of a scale of low to high. And so when they first introduced the virus, it was extremely deadly, upwards of the upper 90s or so percent mortality. But over time, the rabbits began to adapt. They still have, here it says lethality, but effectively mortality. And so it's still deadly. but less so. And so there's a big selection process here where the vast majority of the rabbits die. The ones that don't are slightly less susceptible to that virus. They go on and breed and go through a couple iterations of this, particularly for something like a rabbit that can have 2, maybe 3 litters a year, 6-ish per litter. Short generation time. I'm not sure about rabbits. I know squirrels can breed at 6 to 9 months, depending on how well fed they are. It's likely true for rabbits as well. And so you have a lot of generation times, which means you've got, you're rolling the dice more often genetically, meaning that evolution takes place over generations, not over time. So faster regeneration is the faster evolution will happen. So there's likely some selection here for individuals that are able to survive the myxoma virus, or at least have lower mortality rates. It doesn't mean that they're like totally immune, everything's fine, but the mortality rates are under the 90%. Some plant genotypes have resistance to gene-specific parasite genotypes for gene-to-gene interactions. So wheat has dozens of different genes for resistance to fungi such as wheat rust. Different wheat rust genotypes can overcome different wheat-resistant genotypes. So you have this, you kind of think of it as like, maybe locks and keys is a good analogy. And so wheat could have a series of locks that would keep out this parasitic fungi, these rusts. And then the rusts have a certain number of keys that can unlock those locks. Well, if there's a match between key and lock, then the rust can unlock the wheat, come in and infest. If it doesn't, then the rust doesn't take hold in that wheat. So one wheat can have protection against a number of rusts, and one rust can have the tools to circumvent the protections for a number of different types of wheat protection. And so they can either match or mismatch. This is one looking at snails and the percentage of snails that are infested. And this is with a rates of microphallus. And sorry, I was trying to figure out what the parasite was. I forget what the parasite is on this one. But the infection rate, so here's the snail genotypes, A, B, C, D, E. A through D are the darker ones. Those are the more common genotypes. And the red one is the less common genotype. The percentage of snails infected, particularly for these genotypes, this one's a bit harder to explain away, but nonetheless it is higher than the red. So for these genotypes, they're much more likely to be infected by the parasite compared to the red one. So the idea is that if the parasites are attacking the more common genotype, eventually this genotype will decline and this genotype will increase. And so it's a way of shifting the dominance of certain genotypes, where the parasites are more likely to infest and more likely to cause damage or mortality to a certain genotype. That genotype will eventually be reduced in distribution, and the other genotypes then will have a higher selection and increase in genotypes. So it's a way that parasites can alter the genetic structure of a population by having selective removal, right? And so selective removal is the basis for evolution. If all parasites attacked all genotypes the same, that wouldn't be a genetic selection tool. It would perhaps depress the population, but it would not be acting on an evolutionary scale. Or what this is saying is that because parasites can select for certain genotypes, they're altering the distribution of those genotypes and giving other genotypes a selective advantage. So if this were to persist, you would expect the blue genotypes to decrease and the red genotype to then increase, at least in relative proportion. So it's interesting to think about what's the kind of long-term impact on genetics as it's mediated via parasites. So this ever-escalating arms race can occur. As with snails and trematodes, common host genotypes decrease in frequency because they're attacked by many, leading to an increase of previously rare genotypes. So again, like that snail example, if the parasites are going for the most common genotypes, they're eventually going to drive those genotypes down in distribution, and then the previously rare ones will become both relatively abundant and then probably absolutely abundant, because they're not having that they're not suffering from the parasite, whether that be a mortal infection or just an infection that's robbing you from energy. And as we've seen, a bunch of these parasites also interrupt sexual reproduction. They can be castrating parasites. And this arm race may stop because of trade-offs. So having a defense or a counter defense all comes at a cost. And so, having a defense against a parasite, or as a parasite, having the tools to overcome or defeat the defenses of a host is not free. There is no free lunch. There is no free parasitic defense either. And so because there's some cost of these, and this is often hard to measure, but there's some cost to having a defense against a parasite, or as a parasite having the tools to circumvent a host defense, there must be some cost. There's some energetic cost somewhere. And so eventually this arms race will get just too expensive. See the Cold War if you're not familiar. So in fruit flies and their parasitoids, there's a cost for encapsulation and for avoiding encapsulation. The ability to encapsulate is associated with low larval survival rates. So in the fruit flies that have the ability to encapsulate a parasitoid egg, their larvae have a lower survival rate. So they have this extra tool that can help them defeat a parasite, but generally their offspring are less viable compared to individuals without this adaptation. And wasp eggs that avoid encapsulation by embedding in host tissue take longer to hatch than other eggs. What that means is, the longer it takes for you to hatch, the more likely that fruit fly or your host is likely to die from some other reason. Like if you're an egg, particularly a parasitoid egg, and you're in your host waiting to hatch, and then they get eaten by a bird, it's like, well, game over. So time is always of the essence there. So there's cost to these, and that helps. You can also think of it as a way of mediating, or actually really promoting, the genetic diversity. Whereas in instances where there's no parasites, the individuals without the adaptation to deal with those parasites will do much better. So they'll be genetically more efficient and have more offspring, be more dominant. But then when a parasite comes in, those individuals will be pushed down. Those genotypes will be selected very strongly by the parasite, but then the individual with the adaptation to defeat the parasite, who normally is not as fit, will now rise and become relatively more fit. So it's kind of a way of balancing that out. And then this is one looking at experimental populations of a beetle, looks like a little ladybug, with a sexually transmitted mite parasite. And they monitor them over the next 25 days and the number of eggs laid by the female beetles, finding out that the eggs laid by the female infested with the mite had lower egg production and then much fewer hatching successfully. So the parasite's harming population growth rates. So meaning that what we're looking at here is our control group, just normal beetles. Females are laying their eggs. Between 85 and 100, more or less, percent of them are hatching. In our population where the females are infected with this mite, initially they have pretty good hatching rates, but over time, these females, very few are actually hatching. So we're looking at, often 0, at most in the low 20s. So this is having an impact on their ability to produce offspring for the next generation. So while that individual, may be walking around and still appear relatively healthy, their eggs are now no longer viable. So this would have an overall impact on the growth rate of that population, right? If we went from 90% egg hatching down to 0, that's an issue, right? But even down in the single digits or the teens, it's still happening, but by a lot less. And so that's going to have an impact on the growth rate of that population. The amphipod, Coryphaeum in North Atlantic tidal mudflats can be extremely abundant. 100,000 per meter squared, like that's a lot. So a meter squared is 3 feet by 3 feet. So kind of do like one of these, sort of like that area, having 100,000 of them. And the streamatoid parasite can reduce amphipod populations dramatically. In a 4-month period, this attack from the trematode caused the local extinction of Coryphaeum. I did work on, like I mentioned before, the bean clam in Southern California. And we think that it has this real boom and bust cycle, very classic to a lot of things we've been talking about, where it's not always even, but every 4 to 7 years, there's a massive number of them, and then they go away. And with an undergrad, we did a study looking at the number of trematode cysts in these bean clams. And this little tiny clam, it's centimeter and a half long. They would have 300 plus worms in there. Just really, when you look at the tissue, that's all you're seeing. It was pretty gross. And it was just kind of a little one-off undergrad project. But man, I bet if you would have had more and better data, you could track those populations both through time, the density of them, and then also the parasite load, and see that it was a castrating nematode as well. So it prevented the donax from sexually reproducing. So it was both this massive, just full, tissue just full of these worms that were robbing a ton of energy, as well as preventing them from reproducing. And so I think that's probably what's driving those 4-year cycles. And these 4-year cycles are interesting because they're noted by Coe in 1920 on the same beach. But sadly, there's no data between his work in the 20s and 30s, and then really my work in the 2000s, in the late 2000s. So there used to be a whole bunch of American chestnuts. A fungus came over in the early 1900s and really knocked them out. It's kind of a nasty one that comes in through any wound in the tree, eats the tree from the inside out. It takes about 2 to 10 years to kill the tree, and then their offspring that then come out of a chestnut typically survive for about 10 or 15 years. And so it's one of those ones that's kind of sad because you think at least the babies are coming up, and so there'll be some new hope. But it's not looking good for the chestnut. There's a couple populations around. This is their historic range, and now they're down to a few stands. And there's some hope that there's they'll develop fungi-resistant chestnuts, but likely the patches we see just haven't been knocked out. It's pretty hard for an organism to develop a defense against a totally novel parasite. So if it's not a parasite where they're evolved or co-evolved with a similar parasite, they might just not have the genetic tools in their toolkit to be able to defend themselves against that parasite. This is looking at grouse, red grouse in I think the Northeast, where, similar to my Donax story, where they think that, where I think that Donax cycle every 4 years because of parasite loads. There was an indication that maybe that is what going on with the red grouse, and so they went out and treated a couple populations for this parasite. It's another nematode parasite, and try to see what happened if the treated populations crashed the same way that the untreated ones were. They were arguing this was a big difference. Like this 99 crash is definitely not as extreme. The 93 crash is still there. I mean, that is certainly higher than here. So here's two populations that are controls. One population that was treated once in 1989, some success, and then one that was treated in 89 and 93, some success. So looks like some external factors here as well, not just the disease transmission or being the parasite load from this nematode. Okay, back to more mathematical modeling. There's more terms here. We'll kind of walk through them slowly. So when we're looking at host, part of it's pretty similar to Lacovotera, and so you can think of it in that term as well. So host population is divided into susceptible individuals and infected individuals. So let's just stop there. So susceptible individuals means that every individual in the population who does not have a defense against that parasite. It's typically like all of them, depends on the parasite, but this is all of the individuals that could be a host for this given parasite. Infected individuals are individuals that are hosts, individuals that are carrying that parasite, and then recovered and immune individuals is R. So individuals that have recovered or they're immune to that parasite or R. So either individuals that genetically have a defense against that parasite or individuals that have recovered and have learned how to fend off that parasite. So we have 3 S, susceptible, S, that makes sense. Infected, I, and then they've recovered as R. And so it's often necessary to keep track of both host and pathogen genotypes, and the host age may affect likelihood of infection. We can look at ways of transmission. So latent period is the time in which an individual is infected but cannot spread the disease. This is frequently like an incubation period. In parasites, it could be if that parasite needs to build up enough like in your guts before it starts laying eggs. Like that could take a period of time. In vertical transmissions, when a disease is passed down from mother to newborn, in a simple model, it shows that a disease will spread only if the density of susceptible individuals exceeds a critical threshold density. And so this is saying that you have to have some threshold as a combination of interaction, how often animals bump into each other, how transmissible that disease is, and what the mortality rate looks like. So if you have a high mortality and a low transmission, the disease is likely to go away because everybody that gets it will die, and it's hard for them to pass it on. And so compared to a disease that has a high transmission rate and a relatively low mortality, it's easy to spread around, and it's not killing people. So, once they get it, they're not out of the population. They're still available to be spreaders for quite a while. So S is the density of susceptible individuals. I is the density of infected individuals. And so we're talking about density here because it doesn't really, you could have 100 individuals across, 10,000 kilometers, or you could have 100 individuals across 100 square yards. Those are two different things. And particularly for disease transmission and parasite transmission, it's really proximity that tends to do that. These are generally not large dispersing things. 6 feet might come into play here. So the density of susceptible individuals is S. The density of infected individuals is I. SI is the probability of infected individuals encountering susceptible individuals. And so as density goes up, the probability of those two interacting also goes up. Beta is the transmission coefficient, how effectively the disease spreads. And so if we want to calculate disease transmission, we need to know the density of susceptible and infected individuals. The higher those numbers are, the more likely they are to bump into each other. Just think about, I always think of like blue and red dots. I don't know why, but think about mice, white and black mice in a box together, or diseased and not diseased mice in a box together. The more you put in there, the more they're interacting with one another. And then transmission is how quickly it's able to spread, how easily it's able to spread. So if transmission rates are 100%, every time those two mice bumped into each other, they would spread the disease. If it was 10%, 1 out of every 10 times they bumped into each other, it would spread that disease. I increases when the disease is transmitted successfully, and decreases when infected individuals die or recover. Death and recovery rate is M. So if we have change in the infected individuals over change in time, it's a function of disease transmission, the transmission coefficient, density of susceptible individuals, density of infected individuals. This makes sense. But coming out of the population is the death and recovery rate times the number that are infected. So you are no longer you're no longer one of these two things. You're no longer susceptible, and you're no longer infected if you die. So you're no longer, if you can only be two things, if you can only be somebody who can get the disease and somebody who has the disease. If you die, you're neither of those. And if you recover and then you're immune from the disease, you're no longer susceptible and you're no longer infected. So for this, we don't care if they die or recover, because we're just looking at the change in infection through time, not the change in population through time. We're looking at the change in infection through time. That's what we're modeling. So for this one, M is death and recovery. It seems kind of like funny to lump them together, but for infection transmission, we don't care. You've now left the population of things we care about, because we only care about two things: whether you can be infected and if you're infected. So again, this unit here is the transmission coefficient, susceptibly infected, infected, minus death and recovery rate times the number of infected individuals. And the disease will spread when the change in infection over the change in time is greater than 0. So if it kind of makes sense, every unit time, if there is more infected, than there was before, if it's a positive number, effectively, then it's spreading. This occurs when the transmission coefficient times SI, susceptible and infected, minus recovery and death times infection rate is greater than 0. So let's go back to this here. So if we have, greater than, which one are we at? Greater than 0. Change in infection over change in time. Transmission, susceptibility, infection. This is how many are being, basically this is the unit of how many are being infected, minus how many are being taken out of the infection population. If there's more being taken out of the potential infection population than are being added, you'd have a negative number here. If there are more being infected than are being taken out of the potential infection population, then this number would be positive. This occurs when S, the susceptibility, is greater than the recovery and death rate divided by transmission. And the disease will establish and spread when the number of susceptible individuals exceeds the threshold density ST equals M over B. So I wrote down a way of thinking about S is greater than M over B. I said when the number that die or recover divided by transmission is less than the number that it can spread to. Disease will spread when when the number that die or recover by transmission is less than the number that it can spread to. One way of thinking about that. Disease will establish and spread when the number of susceptible individuals reaches this threshold. This is the threshold of susceptible individuals, which is the mortality and recovery rate divided by the transmission rate. Ways to keep the number of susceptible individuals below the threshold is susceptible domestic animals are sometimes slaughtered to reduce density, especially when the density can spread to humans, such as the bird flu. So they just killed massive numbers of animals. For humans, mass vaccination programs have been successful. And this is the vaccination results. Vaccination reduces the incidence of measles in humans. So this is the number of measles cases in the absolute number, which is interesting because you have to think about the number of people has been increasing, and then the percentage of people vaccinated. So there's a pretty strong relationship between vaccination and reduction in measles. And ways to keep let me scroll over here. Ways to keep the number of susceptible individuals below the threshold is susceptible domestic animals are sometimes... I'm going the wrong direction. Public health measures can raise the ST or the threshold. They increase the recovery rate of infected individuals who then have immunity by early detection and improved treatment, and decrease the transmission rate by quarantining infected individuals or changing behaviors to reduce disease transmission. Another slide says 2001 on it, but this book was written before that. Sort of interesting. determining threshold populations. So this is herd size. Herd with greater than 300 individuals, and then herds with less than 300 individuals, and herds with more than 300 individuals. And the percentage of individuals exposed, there's sort of a high one up here, but this is sorry here. There's number 16 researchers. Threshold densities are determined by populations. Okay, the ST is found to be 200 to 300 per herd, and so yeah, once we're getting, what is this herd size? 200 to 300 ST. I was confused about this one that's way up here. This one's kind of an anomaly. I'm not sure if they explain that anomaly. Oh, okay. There we go. It's here. It's this herd had interacted with a herd of elk that also had the same disease, so they picked it up from somebody else. So when the herd size is less than 300 individuals, the percentage of exposed individuals is quite small, in the single digits. Once it crosses that ST, that threshold, then the percentage of individuals exposed increases quite a lot. These guys caught it from some dirty elk, came by and gave them the disease. And so showing that this is a log plot, so 100, 1,000, 10,000. It's kind of a threshold. It kind of is, I mean, if the difference between 10,000 and 1,900 is really not that much. There's a kind of a small ramp, but it pretty much levels off. That once you get over that threshold, the percentage of individuals that are exposed is kind of the same, which is kind of interesting. So herd sizes in the parks are between 1,000 and 3,000 individuals. Neither option for reducing the ST was feasible. Vaccine wasn't available, and killing large numbers of bisons was not acceptable politically or ecologically. So this has been a conundrum for disease transmission of bison herds in the 6 national parks in the U.S. and Canada. So that's becoming an issue. Okay, we've got one more section here to wrap out Chapter 13.